Thiamethoxam and Clothianidin insecticides lower the Apis mellifera queen's reproductive rate, presented by Courtney Kramer. First, I would like to introduce to you the Western European honeybee, Apis mellifera. The genus is Apis and the species is mellifera. The origin is believed to be somewhere from Asia and Africa. This species functions as a highly developed eusocial superorganism, and it is important and should be studied because they are primary pollinators for plants around the globe. I would like to talk about the basic structure of the reproductive process. Like I mentioned before, the species is a eusocial superorganism, and it has a class that is solely devoted to reproduction. The primary member of this class is the queen because she is the only one within the hive that has advanced reproductive organs, as seen in this picture on the slide. She practices extreme polyandry. This means that she has many mates to provide much genetic code for her offspring. Now I would like to introduce to you two different insecticides, Thiamethoxam and Clothianidin. They were introduced in the 90s. They are neuroactive and they're chemically similar to nicotine. An interesting thing about them is they are water soluble, which means that these insecticides, especially if laced on seeds, can be absorbed into the vascular system of the plant. This means that if a bee or any other insect lands on any part of the plant, the insecticides will be effective. This is why they're so widely used, and another reason why they pose a danger to honeybees. Does an Apis mellifera queen's reproductive capability decline following exposure to Thiamethoxam and Clothianidin insecticides? This experiment studied just that. It looked if Thiamethoxam and Clothianidin reduced the number of Apis mellifera queens laying eggs and their vitality at 4 weeks. The experiment exposed developing honeybee queens to Thiamethoxam and Clothianidin to concentrations expected in the field. It allowed the queens to sexually mature and then observed the production of worker offspring for 4 weeks. Figure A showed that there was a 38% decrease in queens produced workers compared to the control group. And figure B shows that there was a 34% decrease in queens alive and successfully producing worker offspring at 4 weeks compared to the control group. These insecticides severely reduced the number of Apis mellifera queens laying eggs, as well as the number of queens still alive and reproducing at 4 weeks, thus causing a reduction in the number of queen vitality and ultimately affecting their reproduction. This same study also saw or looked to see if there was a reduced amount of spermatozoa and viable sperm in the queens after being exposed to these insecticides. Figure B shows that there was a 20% decrease in stored spermatozoa count, and figure C displays that there was a 9% lower proportion of living sperm versus dead sperm. These insecticides significantly reduce the quality of stored spermatozoa and the proportion of living sperm in Apis mellifera queens, thus reducing the queen's reproductive ability. This same study looked at to see if there was any change in the duration of nuptial flights or mating patterns in the queens. Figure A and B show that after being exposed to these insecticides compared to the control group, that there was really no significant change in the number of flights. And figure C displays that there was no change in mating signs either. They did not alter the duration of flights or the number of mating signs in Apis mellifera queens. From outward appearances, the queen's lost vitality may go unnoticed to human observers. In conclusion, This important study by William et al. Demonstrates the effects caused by the pesticides, thiamethoxam and clothianidin. The effects include a reduction in the number of queens laying eggs, the number of queens alive at 4 weeks, the number of spermatozoa, and the number of live sperm. All of these things affect and decrease fertility in queens, and therefore they could have an overall negative impact on the population of Apis mellifera. Interestingly, these pesticides did not cause outward signs of distress, such as change in flight quantity or duration, or even in mating signs. The significance of this is agricultural use of these pesticides may result in a negative effect and could decline an Apis mellifera population. The queen's decline in reproductive health is not obvious. This may lead to skepticism regarding the significance of the negative effect. A decline in wild populations of Apis mellifera and decreased populations around the globe. Decreased populations result, in turn, cause less forage in the wild, a decline in rangeland health, reduced crop production. Lastly, a serious consequence could be seen in less quality crop production for food security. The effects of thiamethoxam and clothianidin should be brought to the attention of the public and private sector leaders because the fate of our growing population relies on the health and goods honeybees provide through participation in the ecosystem as the primary pollinator. These are the references I used to complete this presentation. Thank you.